This video will cover the topic of Hardy-Weinberg equilibrium, which can be used to measure allele frequencies and changes in allele frequencies in populations. Such changes serve as evidence for evolution and support the big idea that evolution drives life's genetic diversity. It is recommended that you cover Mendelian genetics before watching this video. First, we must define what Hardy-Weinberg equilibrium is. Hardy-Weinberg equilibrium is a state of a population when the following conditions are met. First of all, there must be a large population size. Second, there needs to be no migration. Third, there must be random mating within the population. Fourth, there can be no mutations that affect a change in the population. And fifth, there must be no selection. Now, these 5 criteria are basically what is needed for evolution to occur. So this is a case rarely seen in nature. However, the applications of Hardy-Weinberg equilibrium are widespread. First, we must review some genetics. We know that for each gene in the genome, each individual has 2 alleles. And now we'll call these alleles P and Q. Generally, the allele P indicates dominance, and Q indicates the recessive allele. Since each individual has 2 alleles, there are 3 different possible combinations. There's PP, PQ or QP, and QQ. PP is referred to as the homozygous dominant. PQ or QP is heterozygous, one of each. And QQ is the homozygous recessive. We will use these terms again later in the video. Now let's say we took 5 individuals, and for a single trait we took and gathered together all the alleles, coloring in the P alleles as blue and Q alleles in red. Now for this particular gene pool, the P frequency would be 6/10, or 0.6, and their frequency for Q alleles would be 4/10, or 0.4. As you can see, P plus Q, the frequency for both alleles, always adds up to 1. This will always be true. Now this brings us to what is known as the Hardy-Weinberg equilibrium equation. In this equation, we take the frequencies for all the possibilities in one equation. We have P squared plus 2PQ plus Q squared equals 1. Now let's take a look at the equation. P squared is the homozygous dominant case. 2PQ represents the 2 heterozygous options, and Q squared represents homozygous recessive. The frequencies for all these cases will always add up to 1. And using this equation, we can solve for the different frequencies in a population under Hardy-Weinberg equilibrium. Now let's do an example. Let's say in a population of fruit flies, the red eye allele is recessive to the allele that codes for non-red eyes. If 36% of the population of flies have red eyes, how many are heterozygous? So since we know that the red eye allele is recessive, it'll be signified by Q. And those with red eyes must be homozygous recessive. Thus Q squared is equal to 0.36. By taking the square root, you get that Q would equal 0.6. And then, since we know that P plus Q always equals 1, you can find that P equals 0.4. From the Hardy-Weinberg equation, it says that heterozygous individuals, the frequency is 2pq. Since we have all those values, to find what percentage of the population is heterozygous, you take 2 times the frequency for P, frequency for Q, and you get 0.48. This means that 48% of the population are heterozygous for this trait. Changes in these frequencies over time can prove how evolution impacts a population on a genetic level.